Francis, it's great to see your face on the big screen in the
Ah,
lecture hall. Hi, can you see me?
you're there.
Yeah,
Yeah, I
yeah,
can
I'm
see
here,
you.
of
Yeah.
course, to
Hello.
hosting the session. Yeah, yeah. So I think we should get started. It's already 9 a.m. and I hope you had a good dinner and a good party yesterday and didn't drink too much. So today we're going to continue with the cross-linking session. So we have two speakers in the cross-linking session and then the third speaker before the coffee break. And it's my pleasure to welcome our first speaker of the session today, Francis O'Reilly. So Francis did his PhD in Heidelberg at the EMBL. That's a really prestigious research institute in Europe. Like everyone wants to go there. And then afterwards, he moved to Berlin and joined Yuri Rapsuber's lab as a postdoc. And then afterwards, he moved across continent, so he moved to NIH at National Cancer Institute at the United States. So Francis is not only a cross-linking mass spectrometry expert, and he's one of the few people in the cross-linking field that also has a very strong structural biology background. So he's done crystallography, and he also learned a lot cryo-EM and cryo-ET at the EMBL with Martin Beck. I got to know Francis when he was in Yuri Rapsub's lab in Berlin, but we actually became good colleagues after he moved to NIH. So he's done a lot of great groundbreaking work in cell cross-linking mass spectrometry in combination with AI-based structure prediction, and he continued this work, like in cell cross-linking, look at the native structures of protein complexes and try to look at the protein complex formation and the protein interaction interfaces. And I think this is going to be, or as your main research line at NIH. So today he will talk about structural biology in combination with cross-linking, and we're looking forward to your talk, Francis.
Thank you. So good evening and good morning. So yeah, thank you very much for having me. I will try to speak slowly, as I know my accent is not normal for your ears, I guess, because I'm from Ireland. Okay, so yeah, so today I want to talk about a project that let me just check a timer. Okay, that we had published this year, which is where we worked on protein complexes. And as Fan mentioned, we discovered novel protein complexes inside cells, and then we used Alphafold to help us, along with cross-linking mass spectrometry, to identify the structure of these complexes. Is everything okay, Fan? Can you hear me? Okay.
Oh, good. Oh, good. Oh.
Okay, good. Okay. So what really has driven my career is this very complicated picture of the cell. So a lot of structural biology is done where people purify individual complexes and solve the structure, or they even doing the purification is usually done by tagging a complex and doing affinity pull downs. But things with high off rates are lost in this, are difficult to maintain. It's difficult to maintain these complexes in ex vivo. And also when you're doing structural work, you have to be able to maintain or reconstitute these complexes. And if any of these interactions are mediated by molecular crowding or membrane interactions or DNA and RNA or even things like labile post-translational modifications, this can be very, very difficult. So to get, I think there's a whole array of protein complexes, even on top of what we already know, that are out there yet to be discovered. So when I was working on, as I'll discuss in a minute, we were working on Bacillus subtilis as a model organism, so it's a simple bacteria. When we were doing this, AlphaFold appeared. I thought, my God, can AlphaFold just do all of this work for us? We'd never have to do an experiment again. It can predict all of these novel complexes. And some papers have tried to do large-scale AlphaFold predictions. So here, this Burke et al. From work that was done at EMBL. And what they did was look at all of complexes that were in BioGrid or these other protein interaction databases. And they did an enormous amount of number of predictions, predicting binary pairs of proteins. and then showed that actually AlphaFold was pretty good at predicting these interactions. And then they generated this protein interaction network. The problem, though, with using AlphaFold for predicting all novel complexes is twofold. So on one side is the n-squared problem. So computing a binary pair of interaction with AlphaFold can take, on a GPU, anywhere from 30 minutes up to 8 hours, or even up to 24 hours. And then if you want to do this for all of the protein interactions inside the cell, this could take, for Bacillus subtilis, which only has 4,000 proteins, it could take up to 3,000 years on a single GPU. But that's only binary interactions. If you then want to do trimers or tetramers, these predictions then get into the billions of predictions and could take you millions of years. So obviously this is impractical, but maybe we can use proteomics approaches to actually simplify the problem and then discover novel complexes and use AlphaFold to confirm them. So again, the model organism that we decided to work on was Bacillus subtilis, which is a gram-positive bacteria. And as I said, it only has around 4,000 protein coding genes. We wanted to use the combination of cross-linking mass spectrometry, co-fractionation mass spectrometry, and in combination with Alphafold-Multimer to discover novel complexes and also predict their structures. So it's quite a simple approach when it comes to cross-linking. You just add cross-linker to the bacteria. You have first spin it down from its growth media, resuspend it in PBS, and add DSSO in some DMSO, and then we did, to simplify the cross-link proteome a little bit, we decided to lyse the cells and then do a simple centrifugation step where we had a pellet and then a soluble supernatant, and then also we took some of the soluble supernatant and separated it by size exclusion chromatography. So then we had these 3 data sets, and then we took 20 fractions from the high salt fractions of strong cation exchange, and then we separated them by size exclusion chromatography. So crossing peptides tend to be larger, so then we have this double enrichment step. This became quite a lot of mass spectrometry runs. I think it was hundreds of mass spectrometry runs. I can't remember exactly now. And then after DSSO is cleavable in the mass spectrometer, and it produces these distinctive duplet diagnostic peaks. After we searched all these spectra for crossing peptides, we then went back and checked to see did we have the duplets that represented the 2 peptides that we'd identified, filtered the results on this, and then performed FDR. Okay, so that's quite in-depth proteomics of what we did. But back to the the more biological results. So just to mention that if we did a regular mass spectrometry run of these cells, you can identify about 2,000 proteins in the growth conditions we used. But proteins that we identified with tetrameric crosslinks were only mostly the most abundant 300. So there's a intensity problem with cross-linking mass spectrometry, but nonetheless, with the small number of proteins that we were able to identify in complexes, we still identified quite a few novel interactions. So here you can see the number of crosslinks per PPI. That should be. So let's say we were able to identify 22 crosslinks between these 2 subunits of the pyruvate dehydrogenase complex, and these are in sub-T-wikis. This is a database of known complexes from Bacillus subtilis that's maintained in by interactions that were not in sub-T-wiki. And here these will come up later in the talk, where you see we had quite a few proteins that were in the membrane, which is difficult to identify by other approaches that are not cross-linking mass spectrometry. And then we also had these 2 proteins, UGI and YABR, that we found cross-linked to ribosomal subunits that I'll come back to later in the talk. So if you want to see what these, obviously cross-links are used as distance restraints for structural biology, and just as a kind of a sanity check, we mapped our cross-links onto the RNA polymerase and the ATP synthase, And you see that almost all of these crosslinks fulfilled the distance restraints that we expect from this crosslinker. So we allowed with DSSO that it crosslinks residues that are 30 angstroms apart from C alpha to C alpha. And this is the interaction network that we identified. And what you see is several large known complexes like RNA polymerase here, ATP synthase, DNA gyrase, pyruvate dehydrogenase, and the 50S and 30S ribosome. What you see is the gray lines here are the crosslinks we identified, and the thickness of the gray lines are how many crosslinks we identified between these subunits. And I just want to draw your attention to these two proteins, which are YKUJ and YNER, which are completely uncharacterized. So now this is just the cross-linking mass spec data. We then also wanted to identify additional candidates of complexes by cofractionation mass spectrometry. And here you just separate a proteome by some sort of fractionation technique. Here we did size exclusion chromatography, so separated by size. And the rationale is you take each of the fractions, you subject each of them to quantitative proteomics, and you look at which subunits reproducibly co-elute together. So here you can say that this protein in blue potentially is in two complexes. So what we did was two different experiments. First, we separated our lysate by just our native lysate from Bacillus subtilis without any cross-linking. And here you see the RNA polymerase subunits all summed together, and proteins that are known to bind the RNA polymerase, GRIA and NOSE, elute separately. And that novel interactor that cross-linking identified, YKUJ, also elutes by itself at low molecular weight. But if we added a very small amount of cross-linker to these cells prior to lysing them, you then start to see that these proteins start, a subset of them start joining this complex. So the RNA polymerase shifts from about 500, something like 550 kilodaltons to above 660 kilodaltons in the elution profile. And these proteins, including this novel binder, YKUJ, now co-elute with the RNA polymerase. So we're now confident that YKUJ really is a binder of the RNA polymerase. So we used the cofractionation MS, so proteins that co-eluted by cofractionation MS. This produced about 660 candidate binary interactions. Cross-linking mass spec produces over 200 candidate binary interactions. And then we took all of the binary interactions that have previously been identified mostly by affinity pull downs or by bacterial to hybrid screens that were in this database SubTWiki. We removed all of those that had any structural information, and this left us with 1,200 interactions. And then together, this was over 2,000 that we predicted, and we successfully predicted 1,977 by Alphafold-Multimer. Okay, so at the time, no one else had done a large-scale prediction with Alphafold-Multimer, and we had no idea what the false positives were going to look like. So what we did was plot all of our predictions here in blue. So we did a kind of a target decoy approach where we just look at this predicted interface TM score. I don't have time to get into what this is, but just think of it as a predicted confidence of the interaction of the prediction of the interaction interface. And you see with the bacillus to bacillus predictions, you get this kind of bimodal distribution. And when we then predicted these, what we call decoy predictions, so bacillus subtilis proteins to E. coli proteins, obviously this doesn't happen in nature. We see that we get this other distribution that ends at about 0.85. So then we think that everything with an IPTM above 0.85 is a confident interaction. So if you plot these, so this is IPTM, that interface score that I just mentioned on this axis, and then TM, which is an overall fold score. So if you think about TM as how well the proteins are folded by themselves, and then interface TM, so the IPTM is how well that the Alphafold thinks that the two, how well I think it's predicted the interface. So we just take the IPTMs of 0.85, we see that the majority of the novel interactors are from SubTWiki, but so they're novel complexes, let's say, but they've been suggested before. But if we from our data from cross-linking mass spec and from Coelution, we identified all of these interactors, and we were able to validate all of them by bacterial 2 hybrid afterwards, which is, I think, for those of you that have ever done protein interaction screens, it's kind of an insane hit rate. So we use Alphafold to kind of triage your data. Okay, so this is an example of like it's kind of a panel of some of the interactions that we identified. And here's their IPTM score, so how confident Alphafold is in the interaction, and also then which of our techniques that we identified these. The nice thing about cross-linking is that we identify the interactions by cross-linking, but we don't use the cross-link during the prediction. So we can then map the cross-link onto the cross-links onto the structures afterwards, and show that actually the cross-links agree with where where Alphafold predicts the interface is, which is very nice. So another like neat thing is that because Alphafold does a certain amount of templating, so if it looks into the PDB and sees if there's similar structures in the PDB, it can sometimes trick you into predicting a confident complex that really isn't a complex at all. So these two proteins are orthologous to each other. They have very, very strong sequence homology. So YTOP and YSDC. In the PDB, they're actually predicted as a homodimer, or they are known to be a homodimer. And we actually asked Alphafold to predict them as a heterodimer. Now, Alphafold could do this and just say, look in the PDB and say, there's similar structures there, and fit them both together and say, there you go, I'm confident that this is a complex. Now, it could be wrong because it's done templating off the PDB, but because we have cross-links across the interface and they have sequence information from peptides on both sides, actually, we can now be very confident that this complex really does exist as a heterodimer, not as potentially two different homodimers. And the same with this complex. So there's an example of one of the validation follow-ups we did. So this is YALC, a protein of completely unknown function, bound to ALA-S, which is a alanine tRNA synthetase. And if you're not familiar with these plots, this is a predicted alignment error plot. So it's another score that Alphafold gives you, and it tells you how confident it. So what its predicted alignment error is in angstroms between two sets of residues. So it thinks that all residues from YALC here, it thinks it's very confident in the error. So it's very confident in 3D space relative to each other, where all of these residues are in comparison with this domain. And then it's really not sure how these domains fit together, but it's actually very confident in the individual prediction of this domain and this domain. Okay, that's just an idea how to read these, but it shows you how confident Alphafold is in this interface. And then we did bacterial to hybrid, which worked really, really nicely and showed that that really these do form a nice tight interface inside the cell. Okay, so we had our predicted interactions, and then we wanted to see. We decided could we go bigger from binary interactions. So first of all, I cut the IPTM score at 0.65, so below the very confident 0.85, and we plotted this graph. So you see these are, it's a graph, the clusters made up from binary interaction predictions. You see here we have RNA polymerase, here we have DNA polymerase, etc. And then there's a lot of these proteins actually have transmembrane domains, so we're predicting membrane complexes. So the question is, what do you predict next, right? Do you predict these three as a complex together, these three as a complex together? Maybe you should be predicting two subunits of this, et cetera, et cetera. It actually becomes a giant computational problem because there's so many different, the combinatorics are so huge. So we kind of sidestepped this whole problem. And I'm only showing you half the clusters here because the other half look like this. So there's only 3 proteins in the clusters. So we said, let's go to the next level of complexity, which is predicting these trimers 1 to 1 to 1, right? So one subunit of each. We predicted these by Alphafold again, so we had the graphics card capacity that could at least do this. And again, showing you the same plot as before. This is the IPTM against PTM, and we had a cluster up here of those that had very high scores shown here. So again, we have some like very well-known complexes like pyruvate dehydrogenase with a prediction, very confident prediction to a completely uncharacterized protein here, YNER. So just an aside, here is some of the other complexes that we predict. Here is the 3 subunits of the lactase utilization locus. People didn't know actually that these 3 form one complex together, and we had crosslinks here to confirm that they really do form a complex. But back to YNER, as I discussed. Oops, sorry. That's this protein here, and it's known. So we found it binding to pyruvate PDHA and PDHB, which are 2 subunits of the E1 subunit of pyruvate dehydrogenase, and it's known to form a A2B2 complex, and we had crosslinks that very nicely showed this stoichiometry. We had cofractionation data that showed that YNER really did co-elute with these. Now, bear in mind, only when there was crosslinking involved. There, we had this tiny, tiny bump when there was no crossing involved, just showing the kind of power of using crosslinking to stabilize things prior to cofractionation work. And then in the Alphafold predictions, the top rank, as I mentioned before, we had this templating going on to the PDB. So it looks at the PDB and it finds this distal subunit of pyruvate PDHB and this proximal subunit of PDHA, and it says, this is what I think is the top rank for, and if you're only predicting one complex, you would say, well, this is the prediction Alphafold gives me. But if you go down to rank 7, you start to see this proximal nearby subunit of both PDHB and PDHA. And here is a case where the crosslink that we had to both of these agreed. So we thought this is actually kind of, this is then the interaction that or the complex prediction that we then brought forward. So we now have this, we've now discovered this interaction. We now have a confident Alphafold prediction. And we were then curious, well, can we use the structure to predict what the function of this might be? So YNER binds across the active site of this E1 subunit of pyruvate dehydrogenase. And this is actually the first reaction step happens here, where pyruvate binds into this active site here. So we thought maybe YNER is actually an inhibitor of this complex because it occludes the active site. So what we did was then overexpress this subunit, and if you grow bacteria on glucose, so they don't need pyruvate dehydrogenase, they'll work just fine if YNER, called here PDHI, if YNER is overexpressed. But if you try to grow them only on pyruvate as a carbon source, they then really fail to grow when this is overexpressed. When it's knocked out, they grow just fine. And then for some extra confirmation that this is the interaction that's really important for this phenotype, we look back into the Alphafold predictions. Now, Alphafold predictions, if they're confident enough, can also give you side chain information. And here we saw that there was a tyrosine pointed into the active site, and down here is an arginine that's forming a kind of intricate hydrogen bonding network in the prediction. So we mutated both of these to independently and overexpressed this YNER PDHI, which we now call this pyruvate dehydrogenase inhibitor, so PDHI. We overexpressed both of them and showed that this completely rescued the phenotype. So this was really exciting. Went the entire way from discovery of proteomics through to prediction, through to a known biological function. And then, do I have much time? Maybe another minute or so, just to show that that's not the only stuff that's in this interaction network. Obviously, you can do predictions, but if you have Alphafold can only go so far because we're only doing binary interactions or trinary interactions. And we had proteins also of unknown function, Yabr and Yogi, that had many cross-links to the 30S ribosome. Obviously, with the amount of RNA in here and the number of subunits, we couldn't do AlphaFold predictions, but we were able to work up these with, so the crosslinks kind of suggest, the crosslinks were quite long sometimes here, but they suggest that this, using DisVis, that the interaction space for Yugi is here on the 30S subunit, and for Yabr is down here on the 30S subunit between RS18 and RS6. And if we do sucrose gradients, so here we just lyse the cells and then separate their lysate by sucrose gradients. You can separate at the 30S, 50S, and 70S ribosomes. That Yabr co-elutes with the ribosome with 30S, 50S, and 70S, and Yugi co-elutes with only the 30S, and a non-ribosomal binding protein, ROCF, here as a control, has no co-elution with the ribosome. And also, we could do bacterial to hybrid, suggesting that there really was binding with nearby subunits here. And finally, though we don't fully understand what's going on here, that it's known that tetracycline binds to the ribosome, so it's usually a good control to check if people check to see if the proteins that they're working with can attenuate the effects of tetracycline on the ribosome. And it binds, we know that tetracycline binds are up here nearby RS10. And in this case, when you knock out Yabr down here, when you knock out both Yabr and Yugi, actually proteins have or the cells have a much stronger phenotype while growing on tetracycline. Okay. I will skip this because this is just discussing how we can potentially get to larger complexes with AlphaFold, but it's not really to do with proteomics. And I will finish there, I think. Okay, so just acknowledge that most of this work was done at the Technical University in Berlin in Yuri Rapsiber's lab, along with some like really phenomenal scientists. And oops, sorry, I should. Yeah, so with really some phenomenal scientists, in particular Andrea Graziadei, who is now who runs a core facility in Vienna or in Milan in Italy, and also with our collaborators in Göttingen, who are the real experts at Bacillus subtilis biology. Okay, thank you very much.
Thanks very much, Francis, for the beautiful talk. So any questions from the audience? I think people are a bit shy to come up to the stage because the hand and microphone doesn't work, so they have to come up to ask questions. I will start with one because I actually have two questions to see if I can finish this two, or just ask one. So how do you decide which score you're going to use? Like you use the IPTM score, and we use PLDDT, and I think PAE score does almost have an anti-correlation with the PLDDT score, or as well as IPTM score. So how did you decide, like in the end, which score you use in your score matrix?
Actually, I do have they do very different things. So PLDDT is it's like literally a residue by residue score, right? So it's like how confident is in an individual residue. So let's say this is a prediction of two proteins from ATP synthase that do not actually physically interact. So these two, and here is a case where high score is good, right? So this is the P stands for predicted. So it's very confident of each residue here of the prediction. PAE is an alignment score between, like if you take residue 1 and protein A against residue 2 and protein B, it's saying that it really has no idea where they are in relation to each other, but it knows where all of the ones within protein A are in relation to each other. And you see, this one is an alpha helical. It's very confident of nearby ones because they're nearby in the alpha helix, but any kind of bend or anything, it's not confident. So so that's why you get like this red, so very large alignment errors when you get away from the nearby residues. And then for the TM score, so TM score is an is an overall fold score that's sequence independent. You can kind of imagine it as just taking like a score that gives you this entire square, right, within one protein. But the IPTM score is, you can also imagine that as it's telling you the score of these diagonal ones, where it's saying what is the TM score of protein A to protein B. Do you follow?
Yeah.
So they give you quite different things.
I mean, I kind of know, like, sort of know that what
Sorry.
what they mean, but I don't know like which one to use. Whenever like there's conflicting results with the cross-linking, then I, like which one, like a high score of PLDDT has a low score of PAE with one prediction, and then would you trust it or sort of no?
I mean, it's hard to. I never check PLDDT really when it comes to complexes, because I don't know really. So I've had some amazing examples where you get a terrible IPTM, just terrible, right?
Yeah.
And because here with bacteria, you tend to have like two they're very folded proteins and they form a nice complex together. But when we're working on a lot of human proteins here, you might have everything floppy and a tiny like 10 residue stretch binds to another protein, and you just get like a stripe. You just get a stripe through through here that's blue with a low PAE score. And if you get crosslink nearby there, I think that's pretty good evidence. So
yeah, thanks. I'm waiting for anyone who wants to come up. Please. Yeah.
I want to know how is it evaluate the cross-link information in the model?
Sorry, would you say it again? How?
它是怎么把这个交联信息嵌入到那个模型里面的？嵌入到哪一层？是不是在预测这个 content method 同一层上面的那个？
Hold on for a second, huh? I'm getting the question for you.
I want to know how does it use the cross-link information in predicted the multimer. For the AlphaFold model, he need to use the information to improve the model, right? Or he just used the cross-link tool as a post filter to get the best model. Or which way did he use?
Can you hear the question?
I can't hear, no.
You can't hear. So he's asking you like if you use AlphaLink to improve the model based on the crosslink information you get from the experiments, or you just map the structure onto the crosslink and then, map the crosslink onto the structure and then does a filter to select the best model.
Yeah. That's what we were doing here. I think this is before AlphaLink. So I think AlphaLink now, remember the original AlphaLink paper, I think people don't realize this, is only for monomers. The AlphaLink heteromer one hasn't even come out yet, but it works. Right. I know it works because it's coming out. So, and it works, I think particularly nicely for diazurine-based cross-linking because it's like, tighter distance restraints and things. But I think that's probably the future, to be honest.
Yeah, I think so too. Okay, so then I think the time is out. So thank you so much again, Francis, for joining us today.
Thank you.
And of course, you're very welcome to stay for the next talk in the cross-linking session. But I know it's pretty late for you as well. So, but feel free to just call the end of the day.
So I go off this link and onto the other link, right?
Right. Yeah, I think so. And then, so we'll see each other in person in Hyupo in 2 weeks, and you'll also see many people in the audience at Hyupo.
Oh, I'm excited. I'm excited to see them. I've seen nobody this evening. So
you saw me.
Okay. Yeah, I saw you.
Okay. Then, thanks
all right.
again.
Well, see you all in 2 weeks then. So.
Yeah. Bye.
All right. Bye.